So in this video, we're going to be looking at the photoreceptors themselves for vision. So this is 15B from slide 17. So the photoreceptors, as we saw in the previous video, are the rods and cones. And these cells are specialized. And inside the cells, there's inclusions that are photopigments. And these are molecules that literally physically change shape as they absorb light. So that's found in the outer segment. The inner segment of each goes to the cell body. So it ends up looking like this. So the outer segment has the visual pigments, the things that are going to change shape. The inner segment is involved with the transduction. The cell body of this of this bipolar is here, and then the axon is there. And we'll just kind of leave it at that for now, but they're embedded into the pigmented layer. So the pigment cell holds them in place and has these melanin granules to stop reflection. So that light that hits here and not pass through here and also affect this rod. Yeah, and there's almost like little walls between each rod and cone because of that. So now the rods themselves are very sensitive to dim light. So they're the night vision, they're peripheral vision. But you only see black and white, so it's all gray tones. And there are more rods than there are pathways, so the pathways all end up converging, which then means that you lose visual acuity. You don't perceive the image as distinctly, so it becomes indistinct images. And that's why, as you know, and you're walking in a darkened room with night vision, you don't actually perceive color, and everything is kind of fuzzy. Little Blair Witch Project, you know. So cones are bright light because they're less sensitive, and they're detailed because their pathways are non-converging. Now, each cone has a different pigment. So it's one of 3 different pigments that we have. And then we build the color perception out of which cones are responding how much. And so color is really basically perception out of various mixing and intensity of these 3 different pigments that are in 3 different cones. So there's a couple of different pigments. So retinol is one. It's a light absorbing molecule, and it combines with a protein, right? It's made from vitamin A, and that's why you need vitamin A to see, right? And I'm not going to worry about cis versus trans and what it means physically, but basically cis is bent, trans is straight. So when you go from cis to trans, you end up with some reactions, and that leads to action potentials in the optic nerve. It's rather simplistic, but it's really all that you need to know, is that somehow light causes this this thing to change from its cis form to its trans form, which then sets off a series of reactions that leads to an action potential. So it looks like this, and again, you see it in the membrane. And we'll just leave it at that. So in rods, it's rhodopsin. So it's the cis plus opsin. In the dark, you make rhodopsin. So the trans retinol turns back into cis. You make more of it from vitamin A. When light's absorbed, the rhodopsin breaks down, and the cis is turned to trans. That means the retinol, and so the now trans retinol, separates from the opsin, and so it's really a chemical reaction based on the energy of what's going on. I'm not going to worry about that, but in the dark, it regenerates. Light, it bleaches. Dark, it regenerates. So in cones, it's basically the same thing, except there are different things that respond to different frequencies of light: blue, green, and red. Yellow, will be a mixture of those things. So red and blue, you will perceive purple, etc. Barney taught you that. So if you're missing one of these, then you're going to be colorblind on that, and you won't be able to tell the difference between two colors. So the transduction is how this changes into action potentials. So it's really about opening channels for the cations, sodium and calcium. It causes a depolarization. So in the light, there's a bunch of chemical reactions, and I'm not going to ask you to memorize these, but they end up opening or closing channels. So light, cis, turns into trans, separates, causes a second messenger to make cyclic GMP turn into GMP, which then opens gates, closes gates, and affects the polarization, hyperpolarization, and depolarization. And you can just imagine from there. So these receptors are graded receptors. They don't have action potentials. They have excitatory and inhibitory. So light hyperpolarizes the receptor cells, causing them to stop releasing inhibitory neurotransmitters. So it sounds backwards, but in the dark, there's an inhibitory neurotransmitter, which is glutamate. Right. So you turn that off. The hyperpolarization means that you stop releasing glutamate. Then the bipolar cells are allowed to depolarize and release their neurotransmitters into the ganglion cells, and the ganglion cells are the ones that have the action potentials that go down the optic nerve. So you can follow this along. So in the light, the cyclic gates are closed, you hyperpolarize, right? So no neurotransmitter is released, so a lack of inhibitory neurotransmitters allows the depolarization, which allows a release of neurotransmitter to the ganglion cells, which will activate an action potential and transmit to your brain. So that's the basis of the transduction. Now, in light, large amounts of pigment break down, so you get glare, your pupils constrict, which means that you try to get less light to the retina. So rods cease functioning, right? The cones adapt, and within about 5 minutes, it's working better. And that's light adaptation. Dark adaptation was when you go from bright light to darkness, and it's the reverse. The cones stop functioning, the pupils dilate, and the rods get better. But that takes half an hour or so. That's why you can be very well dark adapted, and if somebody turns on a light, if there's a bright white light, even for a minute, then you will lose your dark adaptation and you have to start over again. So the pathways from the optic nerve pass along the optic nerve to something called the optic chiasma. And this is a desiccation point where some of the fibers go to the left side and some of them go to the right side, right? And then from the chiasma, we have something called the optic tracts, and they go to the thalamus. Then they go to the primary visual cortex in the occipital lobe, right? And there's some visual reflexes, some startling reflexes and things that end up in the superior colliculi in the brainstem. But for the most part, that's not in visual perception. There's some that go to pupillary reflexes, and some of them are for seasonal affect disorder and things like that. So it sets circadian rhythm. But for the most part, we're more worried about visual stimuli. And this is actually the interesting picture. You will see that of the right eye, the lateral side of the right eye and the medial side of the left eye have the same visual field. So the lateral side of the right eye goes to the right occiput. The medial side of the left eye crosses over and goes to the right occiput. So this is looking from underneath the brain. So it's looking up, because this all happens on the underside of the brain. Similarly with the left eye, this area in blue goes to the lateral part of the left eye and the medial part of the right eye. The medial part crosses over. The lateral part stays on the same size in these optic tracts, and they go back here. So that looking straight ahead, both eyes are these. The in here and in here is only one eye. I'm not that worried about that. But the point here is that both eyes are involved with full field of vision, but it separates out here so that this side, the left side, ends up with the right field of vision, and the right side ends up with the left field of vision. So if both eyes are seeing things from slightly different angles, and so we end up with this variation. The further something is away, the less movement happens, or the less difference between left and right. And we perceive the differences in those terms, and we over time learn to translate those perceptions into a depth perception. I'm not going to go over the processing at the different levels, the retinal, the thalamus, and the cortical processing, be it that there's an awful lot of processing that happens in the visual fields, because as a species we rely so much on vision.